other adult out of the house. She can't go. She can't be. She's the one babysitting kids all the time.
You can trust it looks good on TV, but not so cool.
Bright side though, I told her we needed them all yesterday, so I can use the straw. I didn't get a card to buckle. My own. Which I got 2 shirts.
Chapter 12.
I also wanted to tire them out. So. Oh, the Shawnee Mall. I got driving an hour with those kids. They were enjoying it. The 40-minute drive to the Shawnee Mall.
Yeah.
I don't usually. Yeah, it's the closest mall to me. They taste me.
I don't like Shawnee Mall. It's ghetto. I used to do, OBI, we would do blood drive. I don't know, it's just old. It is, it feels old. It is Times Square, like you go there, there's parts of it that feel old, but most people like the fancier area, nice shop, doesn't feel old. Shawnee Mall, man, the whole thing feels old.
What's the number say? The 3638.
That is, oh, sorry, let me move the thing.
Is that the one that?
Get out of the way. There we go, okay.
I don't know, I had like any barely time to study like and stuff.
Yeah.
Thankfully, the pictures are the least of my worries. You wanna know why? I know literally everything else.
Yeah.
So I can bank on. I might struggle with the images because I literally had no time.
Hopefully they'll up, like, 5 minutes.
But I know all the procedures and all that.
Yeah.
So pictures will be the least, my only worry, right? But I'll probably sit outside of the lecture, sit outside and like study them before we get in. So my mom knew my mom knew I had a lot of things to do, but she was like, you're gonna babysit your cousins. I'm like, oh really?
That's not how life works sometimes, yeah, you
Yeah.
can't just like, your cousins are here and drop everything, right?
Yeah, like mom, that's awesome. How about you do it?
Yeah.
Because one of us is trying to get a college education. The other one works at a shell company, no college education. So I go, you take the
Oh my gosh, yeah, I think I might actually remember the 20 as well because of that.
your sister is cross gobbling. Not my sister's cross gobbling, not a bad sister. I got a brother. Oh, the shirt is there. I got it. Yes. I know this one right here. Got like bumpies on it. Like, it's in a velvet. Yeah, it's a function shirt. My brother and I used to wear them all the time, but I would take them to my brother after he was done wearing them. But, I thought it was like a but, my last affliction shirt ripped apart because it was, I think, 25 years old. So I haven't had an affliction shirt since I was like 16 and I missed them. I love them. So I went ahead and went to Buck and bought them. Pressure goes.
Yeah. Yeah.
It's a good purchase. I haven't paid for it. They usually not.
All right, we're gonna get started with the Kahoot in just a second. So our questions at the end of today's lecture is going to be over chapter 15, which is innate immunity. So we're starting to get into the immune system and how it functions. We'll talk about what innate is versus what the acquired immune system is. There's kind of 2 parts to your immunity. We'll get into the talk. But our questions for today, question 1 will be for Jack, who isn't here yet. Question 2 will be Hannah. Question 3 will be Katie. Question 4 will be Reagan. And I'll take care of question 5 when we get to that one, if we have time at the end, of course. Anybody have any questions about the lab exam stuff? Okay, when we go to the lab exam, once we're done with this lecture, I want everybody to try pulling up their lockdown browser stuff and get ready right away. Don't wait till you like we're like in the class getting ready to take the test. We're gonna go into the classroom and you'll be able to get set up and everything. But like as soon as we get in the lab, start pulling up lockdown browser. Don't wait until I'm like, okay, guys, we're gonna take the test now, and I write a pen. Get it set up. I have some paper tests. You must try to access it on your laptop first and show me that it's not working before I give you the paper test. I know there are a lot of my students prefer the paper test. I don't care. Okay. It is way easier for you to get your accurate grade quickly if you do it on the computer. And if I'm making everybody else do it on the computer, unless you have some sort of situation that makes it where you need a paper test, then you must be trying on your computer first. Of course, if your computer doesn't work, then I'm going to give you the paper test. You need to show me that it's not logging in and signing in for you first. And of course, if it craps out on you for your question, then I'll give the paper test so you can just not have to deal with it. But again, come and see me if that's the case. Otherwise, when we get started, we're getting started, and we're going to start the timer. Once I'll write the password on the board, I'll go get situated in my spot, and then we're going to start the timer. There'll be 1 minute at each station, and you must rotate. Not optional if you are going to rotate to the next station. Some of the stations will be like question 34 and 35, and they don't have a microscope at them. It's just like an empty space. So take that time on those questions. Like half of the test is that type of a question, just station, where it's no microscope or something to look at for reference to. Answer any of the questions that are not microscope questions. All right, just take the time to answer those questions. You can go in the order of your station if you want to, but if you wind yourself up and you've rotated into a microscope question, find that microscope question's number and answer that question while you're there. Focus on the microscope question when you're at the microscope. Does that make sense? Okay, so if you get any questions about it during the test, absolutely ask questions. If you have questions about the wording on anything on the test, ask questions, because probably a lot of people will benefit from that sort of stuff. So you need clarification. If you're not sure which station you're going to or whatever, we'll go over which stations and how to rotate. There's an arrow at every single station that tells you where to move next. Actually pay attention to that, and it'll save you a lot of headache. There's always people that get flustered from the test. And they're like, oh my God, I don't know which station I go to next, when there's like a move that's not immediately next to them. So obviously you can't rotate through all those things and not have to make a big jump now. And it says on the arrow where you're supposed to go to. Get flustered, you can ask me. I'm there to help you with that. That part shouldn't be the stressful part. The rotating part shouldn't be the stressful part. What you guys should be worrying about is making sure that you're answering your questions correctly. That's what you should be focusing on. All right. My recommendation, you study your lab stuff however you want to, but once we're done with this lecture, everybody should be focusing on bringing up their lockdown browser to get ready to take the test. And maybe before you do that, if you have some time before we start the test, maybe review your recommendations on how to deal with test anxiety. Don't try to cram right before the test. It's just going to make it worse. That was one of my recommendations on dealing with test anxiety that I'm sure none of you guys are actually paying attention to. When we say don't cram the night before and don't cram right before the test, that's because when you start looking at those words when you're stressed out, it starts to make less sense to you almost instantly because your brain isn't thinking through it logically, it's thinking through it in stress mode. And so then you'll start reading content that may have made perfect sense to you when we're sitting here talking about it conversationally. And then on the test, now suddenly you're overthinking things. Oh my God, it said this thing. I didn't read it all the way through. What did it say? Oh my God, I think it said this. Oh, what if she meant this? She mentioned this one thing this one time. Stop. I don't want anybody reviewing their notes right before the exam. Don't do it. You can do it. I'm not going to come in there, slap your wrist or something. But good God, you guys, prepare before the test. Not immediately the seconds before the test, before the test. That's when you're supposed to be preparing for tests, not the night before. I know all of you wait until the night before the test, that's what you do. And I can't tell you that I can make you live your lives differently. There's a reason that people tell you this shit. It's not for my benefit, it's for your benefit. I don't give a crap if you cram the night before and do shit on my test. I'm trying to help you do better on your test. If you don't want to do better, fine, cram the night before. Don't let me stop you. But I'm telling you for a reason. Science proves if you actually take the time to learn the material over time, you'll do better on the test rather than waiting until the night before to cram. You're relying on one Kahoot and going over these Kahoot questions over and over and over again. Do you actually even understand why the answers are the answers or why the wrong answers are wrong? Do you even understand that? Because if you don't, you're not going to do good on this test. I don't know what people expect. They go to classes and they're prepping for tests In so many classes, you'll go take that class, they'll do like a review the night before, and every single question on the test is literally straight from that review. Shame on those professors. Are you actually learning that stuff? The whole point of college is to help you learn how to learn, as well as to actually learn this information well enough to apply it. If you're just memorizing for the sake of taking that one test to check that box, shame on you at that point. That's not what college is for. It's supposed to be learning, not just passing the test. That's not what college is for. You're not getting that, and I don't know how to help you because, like I said, I am not exaggerating when I tell you nursing school is way harder than this class. And it is none of the classes that you'll take in nursing school will be easier than this class. None of them. None. I don't mean you'll have some hard classes. I mean, all of nursing school will be harder than this class. So if you are struggling in this class to learn how to learn in this class, get ready, man. It doesn't get easier from here. You'll be lucky to get the support that you got from me, from any of your professors in nursing school as well. Because regularly I have students come complain, and they'll just like tell you, like, we went over this in class, you're supposed to know this, and that's their answer when you come to them for questions. Get ready, because it doesn't get easier. Trying to help you learn how to learn. Take this as an opportunity to learn how to learn, so that when you get to nursing school, that stuff won't be as hard on you, okay? So this is midterm, that's why I'm bringing this up, because we still got, of course, the lab midterm, there's not much more we can do to prep for that, right? But we've got Unit 3 exam, we've got the Unit 4 exam, which is half comprehensive, and we've got the lab final. Those are 3 massive tests. Your lab final is worth more than your lab midterm because it's a little bit comprehensive. The actual final exam, which is our Unit 4 exam, half Unit 4, half Units 1, 2, and 3. 100 questions. It's worth more. Those 2 exams by themselves are 20% of your grade. So if you're not actually retaining this information and it's not making sense to you, like if you went through Unit 1 and then you dropped it to move on to Unit 2, every unit that we do builds off of the previous units. I'm sure you're starting to get that a little bit now with Unit 2. We're talking a lot about gram positives and gram negatives. We're talking about the cell wall and how the drugs affect them in Unit 3 and things like this. Like we're picking up what we learned in the other units and moving on with it. Unit 4 is entirely taking all of Units 1, 2, and 3 and applying them to the diseases. So if you're not getting it from Units 1, 2, and 3, the course is set up the way that it is, but you're meant to retain that information, not learn it and then set it down and never look at it again. Guaranteed on your first day of nursing school that you're going to have microbiology stuff. Guaranteed. Almost every single student that actually tries in my class will come and tell me, yep, we have micro almost every single day. There's some aspect of what we learned in microbiology. I can't tell you how many times you're going to talk about C. diff in freaking nursing school. It's ridiculous. There's a reason why I bring it up a lot in class is because we're going to talk about it in nursing school. And if you're sitting there like, what was C. diff again? Guys, come on, right? So take that into consideration as you move forward. Whatever you've done to prep for the lab midterm, take the time between now and when you're actually taking the lab midterm to set your mind into the right mode. Don't sit there and try to cram at the last second, like literally. Take that time to set your mind into logic application mode, not into freak out and cram for the last second mode. We at least agree on that? Because if you waited until right before the test to start like looking at your notes and hopefully you'll learn some stuff, you're not going to do well. Doesn't matter what you do. All right, so let's go ahead and get going with our Kahoot. Chapter 12. So these questions, reviewing chapter 12 stuff. What is selective toxicity? So this was the concept side of things. We talked about selective toxicity and therapeutic index, and they're pretty similar, except for a couple of things. Selective toxicity is the concept that you're wanting drugs to be harmful to the bad guy or work against whatever you're trying to treat. You want to be as effective against what you're trying to treat without being toxic to your patient. That's the concept of selective toxicity. Therapeutic index is taking the actual numbers and applying it to that. So at what point is it going to cause damage to the microbe? At this amount. At what point does it cause damage to the host? At this amount. The ratio between those is therapeutic index. The space between those, the difference. So if we said for humans, it becomes toxic at 1 milligram. For microbes, it starts to work at 0.2. That difference of 0.8 in between them is called the therapeutic window. Okay. All right, moving on. Question 2, select all that apply. What types of organisms can antibiotics treat? Antibiotics. Antibiotics only treat bacteria. Antifungals treat fungi. Antiprotozoals treat protozoa. Anti-helminths treat helminths, and virals treat viruses. Antibiotics only treat bacteria. It's the only one that it's not in the name. All right, number 3, multi-select, select all that apply. Which antibiotics in the list affect the cell wall? These questions are pretty similar to what you'll see on the Unit 3 exam for Chapter 12. And of course, on the unit exams, you'll only have one choice, right? That'll be right. But this one, we get multi-select, so I'm going to challenge your thinking. So obviously the cillins, I'm glad that most of us are getting the cillins on this one. The cillins obviously affect the cell wall. We talk about it a lot. But carbapenem also falls into that category. One of these will probably be choices later on, but ciprofloxacin affects DNA and RNA synthesis. Ketoconazole is an antifungal. That's all you have to know about it. You don't need to know how it works. Just antifungal. Put it in that. Next question. Which of the following affect the ribosomes or protein production? All right, so this is erythromycin. So generally speaking, mycins affect the ribosomal protein production, generally speaking. Vancomycin is the only deviant there. So what, does anybody remember what vancomycin affects? It affects the cell wall of bacteria. So just like the cillins, okay? So vancomycin is the deviant. Other mycins typically are going to be associated with affecting the ribosomes. This will include our larger groups of our aminoglycosides and our macrolides, and then the cyclins like tetracycline and glycylcycline. So those are the groups that'll be in the protein one. But if it ends in mycin, or if I just say aminoglycoside, or if I say tetracycline or glycylcycline, definitely those protein production. Mycins, the only deviant is vancomycin. All right, next one. You may select more than one on this one as well. Which antibiotics affect fulvic acid synthesis? So this one is trimethoprim and silver sulfadiazine. So the sulfas, and trimethoprim is in that category as well. Usually we'll use trimethoprim in conjunction with another drug, sulfamethoxazole, and that's Bactrim, but trimethoprim still falls in this category, okay? So trimethoprim and silver sulfadiazine. The sulfa, yeah. This yeah, stinky. I think of trimethoprim as being like tri being 3 and metho being like methanes. And so there's 3 methanes. Man, that must be really stinky. And that's how I lump it in with the sulfurs. You do what works for you, man. All right, number 6, which antibiotic affects the cell membrane? You guys think I don't still use mnemonics like that to remember the stuff I'm teaching you? That's how I remember it. It's a lot to retain. So which antibiotics of this list affect the cell membrane? Not the wall, the membrane. All right, so this one is just polymyxin. Vancomycin, yeah, that was why I wanted to clarify it after some people had already answered, but that was the cell membrane. So the cell wall was the cillins, and then we had some of our outliers of our cephalosporins, carbapenems, bacitracin, vancomycin. Those guys are cell wall guys. All right, ciprofloxacin, again, that's for DNA RNA synthesis. Polymyxin is cell membrane. So it works pretty similarly to our surfactants and detergents. It mimics your phospholipids. Which of the following antibiotics affect nucleic acids? DNA and RNA synthesis for this one. These are definitely, when you guys get to studying for Unit 3, Chapter 12 and Chapter 11, I feel like, are very flashcard friendly. Great. This is all about memorizing which ones fall into which categories. So this one is Cipro. So anytime it's floxacin, those are the only ones that I put on the test for the nucleic acid ones will be ones that end in floxacin. And you'll have to know that floxacins fall under a bigger category called fluoroquinolones. The fluoroquinolones work by affecting nucleic acid synthesis. Okay, so the floxacins for this one. Metronidazole is kind of a special category drug all on its own. Sort of like our vancomycin was the deviant for the cell wall stuff. Metronidazole is a deviant as well. We're going to have it in a little bit. Which of the following antivirals is used to treat the flu? It should be easy.
I pressed the wrong one.
That's okay. That's why I don't like to use the Kahoots for the actual grade. It's easy to press the wrong one, or you can't go back and change your
I'm like, I
answer.
will crash.
Yeah, I don't like it. So try to steer away from that. All righty, so which one's used for the flu? Yeah, it's in the name. It's just Tamiflu. So penicillin, bacterial cell walls, acyclovir. Acyclovir is an antiviral. Vir in there hints at that. So it is an antiviral, but it doesn't treat the flu. It treats, typically we use that for treating herpes or other DNA-based viruses. We didn't get into the details of how it works, but acyclovir is also known as valcyclovir, which is also known as Valtrex. So acyclovir and Valtrex are basically the same.
Is there a virus that affects DNA called provirus?
What?
A provirus?
If it's inserted into your DNA, then it becomes known as a provirus. Yes. Really good connection there. Something we don't talk about a lot. All right, which of the following is an antifungal? Select all that apply. I feel like there's two of these that you should definitely not be selecting after we just had the discussion on the last slide. Alrighty, so the other two, amphotericin B and miconazole. I'm glad that we're picking up on the miconazole. Miconazole, this is one of the azoles. And in general, azoles are antifungals. There was one deviant, just like with the mycin, the one deviant was vancomycin, it affects the cell wall. There is an azole that we had to learn. Do you guys remember which azole it was that was not an antifungal?
No, ma'am.
Metronidazole. So metronidazole, and the reason I remember it is, it grew up, went to the big metro and moved on to treating more than one thing. So that to me is like, I don't know, however you want to remember it, but that's how I think of them. It, moved on to the big city.
It's a broad one.
It graduated or whatever. So metronidazole is a little more broad. Metronidazole is used for anaerobic bacteria as well as protozoa. Right? So it's typically what metronidazole is for. But miconazole, all the other azoles, antifungals. Amphotericin B is just an extra one that we need to remember to lump into the antifungals.
You said protozoa and what?
Anaerobic bacteria, the ones that can't do oxygen. That may or may not be the next question, FYI. What is a commonly used antiprotozoal that can also be used for anaerobic bacteria? I just said it. Let's see if we were paying attention. Okay, so that's metronidazole. Ivermectin is used for helminth infection. So helminths are also eukaryotes, but they are very vastly different from the protozoa. So helminths are actually animals, or multicellular organisms. They have, sexual reproduction. They lay eggs, things like that. So the helminths are our parasitic worms. They're actual animals. They're classified as animals.
They're the only
Protozoa, huh?
They're the only ones that can lay eggs.
Yes, they're the only ones that can lay eggs. And so in our microbiology course, right? So their helminths are ivermectin, praziquantel, pyrantel, and those drugs there. So both ivermectin and praziquantel, helminth, anti-helminth. Chloroquinoline, I don't know, that's kind of made up. Chloroquine is a drug that's based off of our quinine, and it's used to treat malaria. So that is an antiprotozoal because malaria is a protozoa, but it's not a drug that is used for anaerobic bacteria. It's called chloroquine. It's not called chloroquinoline. That's a made up thing. So I won't have BS that looks like the word, on the exam because I don't feel like that's fair. So I feel like recognizing drugs from other drugs that you should also know is more fair. So FYI, but metronidazole, we use this to treat amoeba infections as well as anaerobic bacterial infections. So it's the azole that moved on to the big city, Metro. All right, so that's it. So let's look at our podium and we're going to move into chapter 15. Just getting into the immune system, which is my favorite. I think that the metabolism stuff is super cool in the grand scheme of things, but I also feel like with the immune system, it's severely undertaught in biology. Most people don't understand how their immune system works. They just know that they have one and that it fights the bad guys, but they don't know how. If you're going into medicine, I feel like it helps a lot to understand that stuff. So, moving on. Come on. Sleep, good job. Let's go. Chapter 15. Alrighty. No, of course I didn't. I left it in the other room. All right. The host defenses. So we start getting into the immune system and how your body fights things off. We have innate immunity, which is like built in. It's not specific, but it's your built-in mechanisms to help with fighting off bad guys and preventing infection. Then we have acquired immunity. Acquired immunity is the one where we get memory, and it's where we're going to have an immune attack to a specific pathogen. And so acquired immunity is what we'll be talking about in chapter 16, but innate immunity we're going to talk about in this. So innate immunity includes two sides, your first line of defenses and your second line of defenses. Your first line of defenses refers to like physical barriers, the acid in your stomach, things like this, that obviously like they have other functions in your body, but it also can work to prevent microbes from getting access to your body. So saliva has an enzyme in it called lysozyme. Lysozyme is an enzyme that breaks down peptidoglycan. That's what it does. That's its function. And so the fact that we have that enzyme in your saliva should be a hint to you that already you have enzymes that are there at your very first interactions with microbes that aren't part of your like immune cells, but have functions with preventing microbes from getting in. So the fact that you have keratinized skin cells, that you have nails, that you have all these things to protect from physical damage, but also to protect from microbes being able to get in. If we burn off your outer layer of your epidermis, that outer layer, if you have a 2nd degree burn or a 3rd degree burn, you have susceptibility to microbes getting into your skin, right? Because you need that outer layer that's all working together to prevent microbes from getting into the deeper layers. So that's already an example of a physical barrier. We also have our microbiome, all of our good microbes that are taking up those stadium seeds and making anti like bacterial peptides and things like that that prevent the bad guys from being able to take hold, set up shop, start the infections, right? So the fact that they're there takes up the space. The fact that they can make antimicrobial things to prevent other things from growing, and the fact they're using up the nutrients in the area, all can prevent our pathogens from getting hold. And when I talk to you guys about how ubiquitous microorganisms are, and we looked at how nasty the bottoms of our shoes were, if you were to lick the bottom of your shoe, what are the odds that you're really going to get sick from it? It's actually pretty low, right? The odds that you'll actually get sick and have an infection from that. Because we have all these barriers in place to deal with those thousands and thousands of microbes that you're actually going to be coming in contact with on a daily basis. In fact, I come up here and type on this and I'm going to touch my face. I touch my face a lot. None of this stuff is sterile. Stuff that's in the air. If you guys are coughing and sneezing, or if you're just talking and releasing microbes just from your lungs or from your throat or what are your sinuses, all of this stuff. The fact that whenever you go to the bathroom and you're touching a faucet, the fact that all of this stuff that we're coming in contact all the time, a lot of this stuff is controlled from your innate system, and some of it will be dealt with your acquired system. It's when things get past the innate system that you're going to need your acquired, right? So that's what the innate system is about. It's about preventing the infection in the first place. And if you get the infection, our second line of defense, which is our initial white blood cells, our phagocytes, the things that are just going to eat up something that's bad. they're not really doing it very specifically. They're just like, oh, you're not self. Yeah, I'll eat you up. Or you're a dead cell. You can't tell me that you're alive. I'll eat you up so we can digest you and break you down and use your parts. They're also going to signal your acquired system to kick into gear and start reacting. So that's how all this stuff is going to work together. And we're going to talk about the functions of each. Immunology is basically the study of how your immune system is dealing with these foreign things. All of your immune system is going to be involved in surveilling your body constantly for foreign things, recognizing it as foreign or self, and then destroying anything that is foreign. So a lot of this stuff depends on recognizing self versus non-self. So there are markers, antigens. Anytime there's a molecule on the surface of the cell, your body potentially could interact with it somehow. And therefore that makes it an antigen. And so any marker that is seen as non-self, we would call an antigen. So these are markers that help us identify self versus non-self. And so you might be saying, okay, so how do we know that it's self? Well, there are molecules on the surface of all of our cells that if they are present, our immune system has been trained during your development from being a baby up into adulthood, that basically it's constantly training itself as to what you should react to as foreign and what you shouldn't react to, right? And what's self. So you shouldn't be attacking self. And from birth, your cells are getting trained into what's self and what you shouldn't react to, and what's foreign and what you should react to. And a lot of that training actually is helped by your gut biome. So your gut biome and the stuff that's there, your body has to know that that stuff's supposed to be in your gut. Otherwise, you'd be attacking it constantly because it's bacteria. And so your gut has to know that that stuff belongs in the gut. But your body also has to know that if it's not in the gut, it doesn't belong in your bloodstream. So your body has to know those sorts of differences, and you're constantly being trained on this stuff based on markers that are on the cell. So literal protein molecules that your body can just say, oh, if this is on this cell, I've seen this before, I've been told that this means self. And so it got trained at some point through its development of all these cells in order to recognize self versus non-self. And all of your immune system cells are involved in analyzing that constantly. Sometimes these signals get mixed up. If you start seeing something that is self and you start recognizing it as foreign, then that can lead to an autoimmune disease. So if it's supposed to be self and your body comes up and says, oh, you're a bad guy, I'm going to attack you now, but it's self, it's your own cells, that's autoimmune disease. So this stuff has to be regulated and your body has to be trained to behave appropriately, but this doesn't always work perfectly. And there are scenarios that can skew it toward wrong. So if a bacteria comes into your body and it has molecules that look like self, in addition to bacterial molecules, then your body can get confused with the signals that are going on there to say, oh, this is bad guy, attack it, and accidentally say, oh, this is bad guy, attack it to the thing that looks like self. And now suddenly you're attacking self in addition to the microbe. So that often can be what can trigger autoimmune diseases. So an example of an autoimmune disease would be rheumatoid arthritis. Rheumatoid arthritis basically is your immune system attacking molecules that are in your joints and your connective tissues and things like that. And then we get inflammation in those joints. That inflammation can lead to building up of bone, calcification in those areas. We have swelling of the joints and then start to lose function in those joints as well. And that can all be triggered just from having seen some of these microbes in your life at some point and recognize them accidentally and recognize parts of them as foreign, as they were foreign, but it matches up with molecules in your joints and things like that that are self. And now we're attacking those self molecules. Now there's been a lot of research into trying to figure out which microbes and what parts of the microbes might be triggering this effect to try to study more and understand it more so we can prevent it in the future, but we still have a very limited understanding of how this stuff works. This is way bigger picture than how I'm describing it, but that's the gist of it, right? There are some molecules that your body can see and know this molecule doesn't belong here at all. And these are PAMPs. So these are pathogen-associated molecular patterns, and you better believe that's going to be on the Unit 3 test, you being able to know what that acronym means. Pathogen-associated molecular patterns. So they're particular molecules that have patterns that we can just automatically say this is a pathogen because it has this. So some examples of PAMPs would be LPS, lipopolysaccharide, which is endotoxin, right? So that is one of the PAMPs. So if that's in your bloodstream, your body automatically knows this doesn't belong in here. we need to start attacking. So that's a PAM. Is that a specific interaction to one specific microbe? No, because any of your gram negatives have that. So that's not a specific acquired immunity response. This is an innate response. This is a wide group of microbes that would have that PAM. So PAMs are associated with the innate response. They're automatic. If you see this, it means a group of pathogens attack it automatically. So it's not a specific reaction. It's a general group reaction. So LPS, peptidoglycan, also shouldn't be in your bloodstream, for example. The fact that there is peptidoglycan hanging around is a concern for your body. Another one is double-stranded RNA. Nowhere in our body do we actually have two strands of RNA working together. Even your tRNA, it's just one strand of RNA folded up on itself. But if you have two separate strands of RNA bound together, that's something that viruses can have as a genome. So that's an automatic indication that if there's double-stranded RNA, that's a PAM that lets your body know that there's a possible viral infection. So there's some obvious ones that we can target whole groups of microbes with. There's other ones too that you don't need to worry about, but that's the idea of what a PAM is. And we have these receptors called pattern recognition receptors that recognize PAMs. So the PRRs are special receptors that recognize PAMs. I won't test you on the PRRs, okay? But you need to know what a PAM is. It's just a molecular group that if we see it, the RNA system knows immediately that this is foreign. We don't have to worry about self-markers here. If we see a PAM, it immediately triggers, this is foreign, time to attack. All right. Your lymphatic system. Again, another part of the system of the body that I don't know why people are just like, your lymphatic system. You know, it's basically just your immune response. And that's like all we learn about it up until physiology or maybe a little bit in anatomy. You'll learn like what a lymph node is a little bit, but I feel like this is a really underappreciated area of your body. So the lymphatic system, it's essentially when let's say you stub your toe, okay, and it hurts. So you've got some nerve signals that are getting sent, they're sending pain, letting your body know that there was some sort of injury response going on. That's important. You need to know sometimes that you have something in some part of your body where you've got some sort of injury response going on. So that's your body's way of letting you know that you've been exposed to something that could potentially be injurious. That pain has purpose. In fact, if you study people that have no pain response, which these people do exist, not just in the movie Novocaine, it's also like actual thing. You can be born with the inability to feel pain. These people are at extreme risk for serious injury in their life. So this is one of the situations where you're cooking and somebody comes and talks up to you and you like lean like this, and you might be hanging out on the stove. The fact that you can feel the heat might be one thing, but the pain from the burning, which you would get almost instantly if you touched a hot burner, right? It would hurt and you pull away from the pain. The heat isn't what's indicating to you that you're getting burned. It's the pain response. These people don't have any pain response. They wouldn't even know they're getting hurt. It might feel warm, but they might not be thinking about it because they're not feeling pain that signals them to they just burn right through their whole flesh and they won't have pain associated with that. No pain at all for those people. Pain is there for a reason. We don't like it, but it tells your body that there's something injurious going on. Imagine that you get in a car wreck and you have your head caved in, and let's say that you had a head injury that was internal. There's no indication of any bleeding, there might be minor bleeding or something like that. But if you had no pain to let you know that you had some part of your body injured, how would you even know that you got injured? Right, so there's serious risk with people who do not feel pain in that sort of department, especially internal injuries, things like that. So pain is there for a reason. So let's say you stub your toe and yeah, you feel the pain and it lets you know there's an injury response. So what often happens whenever we injure ourselves? One of the first responses that we see is redness and swelling, right? So what happens is your blood is flowing to that area. Your body, from that injury response, gets signals to send blood to that area. Why? Why do you need blood sent to that area? So we can start repairing, but really what's going on there? What's actually involved in that process, right? So we have molecules that are in our blood that need to get to that area that are involved in the repair process, like platelets and things like that to clot bleeding. But we also have the bringing in of, that stuff doesn't just move in there on its own as an individual molecule. It comes in there with the fluid. The fluid is helping move those molecules in there. And so the fluid will leave your blood vessels and go into those tissues and start moving the molecules that come with it, as well as your white blood cells that might be involved in infection and cells that are involved in the inflammatory response. They're all moving in there out of your bloodstream in the fluid from your blood into those areas. And so that's why a lot of times whenever you get injured and you have that swelling that's going on, that fluid is moving out of your bloodstream with all the proteins and the good white blood cells and everything with it. That leads to swelling. And why sometimes whenever you have an injury, you'll see like that clear fluid. That's literally the fluid from your bloodstream, right? This all has that exudate, what we might call it. It's exuding out of your blood vessels, moving into those tissues with all the signals for the response to that injury. So we have movement out of that bloodstream into that area. Well, let's say you hit your toe, you stub your toe, you got the fluid moving in that area. We're starting with our inflammatory response. It turns red because blood is flowing to that area. It turns warm because blood is warm and it's flowed into that area. It's swelled because the fluid is moving out of your blood vessels into that tissue. So we've got pain, we've got swelling, we've got redness, we've got all this stuff going on associated with that injury response. And we've got the warmth as well. So those are like the 4 indicators of inflammation, which we're going to talk about in a moment, and we'll get into the terms for that. There's Latin words for all of that. If you speak Spanish, it's going to be easy for you. But anyways, so that fluid is moving out of your bloodstream and carrying all of that good repairing stuff with it. All the proteins for signaling for repair, all the white blood cells to eat up anything foreign or dead cells that might have gotten killed in that injury. All that stuff's happening by moving it out of the bloodstream. So when that fluid is now in that compartment in your stubbed toe, there could be microbes from that area that your white blood cells ate up. And maybe they didn't catch some of those microbes. So they're just hanging out there in that swollen area until your immune system can deal with it. So if we want to get that fluid, it doesn't stay there, right? If you stub your toe and you get a swollen toe, it swells up for like a couple of days, right? And then gradually that swelling will go down on its own. Where does that fluid go? It goes into your lymphatic system. It does, and then it'll eventually go back into your bloodstream. But first, it goes into your lymphatic system. So anytime you have fluid rushing out into an area of potential injury or infection, that fluid comes back first through the lymphatic system. So the lymphatic system is a drainage system from those tissue injury areas or infection areas. That fluid comes back into the lymphatic system, not into your bloodstream. It always goes into the lymphatic system first. Why? So the reason that it goes into your lymphatic system first is that your lymph nodes and all of your good white blood cells, like your B cells and your phagocytes and your T cells, all this stuff will be hanging out in your lymphatic system and in your lymph nodes. It's basically like a screening point. And I mean, I'm telling you, you have lymph nodes everywhere. You don't just have a couple, you don't just have them here and in your armpits and some in your gut. I mean, I'm talking about you have thousands of lymph nodes throughout your whole body, and some of them are bigger than others, and some of them you'll notice more than others because they'll be closer to the surface of your skin. And so those guys, basically what they are checkpoints. And so imagine that they are like a border patrol. So if you're driving through Europe and you're going through different countries, you might have to stop for each country and show that you have papers to keep continuing into the other country. So imagine it like that your lymph nodes are checkpoints and saying everything that's coming through your lymph has to check first. We have to make sure it has clearance first. And that's where we're going to catch our pathogens that didn't get eaten up by our white blood cells, or it's where we're going to get white blood cells that are holding up a flag saying, I caught something, I caught something. That's where we're going to interact with those guys yelling out, I caught something, you need to check this out. I saw a PAMP. I saw, LPS. Come check out what I picked up and make sure that we need to, if we have to have an immune response to this. And so your white blood cells in your lymph nodes are basically looking out for the flags, or they're looking out for the microbes themselves. So everything has to be filtered through your lymphatic system. So before your lymph, your fluid from your infection area or your injury area can rejoin your bloodstream, it has to be screened. And it'll be screened multiple times in multiple lymph nodes all the way on its journey back. And it rejoins your bloodstream once we get towards the heart. So lymph only flows from extremities to the heart. Lymph is also not pumped by the heart because we're not actually pumping it out. It's not being pumped back in. So how does lymph move from your extremities into the heart? Do you guys know how that is managed? By skeletal muscles. Your lymph will just sit there in your extremity unless you move around. And so the movement of your skeletal muscles. Do you guys ever wonder why when you have surgery, or if you know somebody that had surgery, they put those things on their legs that like squeeze the legs alternately? Mm-hmm. Now you know. And to get their lymph flowing. And so if you are injured and you're immobile, One of the number one things that medicine will tell you is the first chance you get your ass up and start moving. Why? Because your lymph can't cycle through and get rid of everything. It can't screen anything. Nothing's moving through the checkpoint at all unless you're pumping it with your skeletal muscles. So in order to filter out everything from your extremities and where you see infection, everything like that, that fluid can't get filtered unless you're moving it with your skeletal muscles. So somebody who is paralyzed, we have to actually physically move their body to help their lymph flow better. Yes.
Is that the reason that once you're shown with being overweight, you have a higher chance of developing lymphedema?
Issues with lymphedema associated with having higher weight are usually associated with risk for diabetes.
Yeah.
And so risk with diabetes itself comes with a risk of poor circulation. So we can't even get the fluid to those compartments to start the swelling process. And then once the fluid gets to that, moving fluid out of those compartments, even with skeletal muscle movement, there's still restriction of the vessels of the lymph as well as the blood vessels. And so getting it out of those areas is hard to clear that fluid. So if anything dealing with movement of fluid in vessels, whether it's circulatory or lymphatic vessels, and they're separate vessels, okay? So anything dealing with movement of fluid in vessels is going to be decreased because of what's going on in your anatomy due to risk with type 2 diabetes. And so you could even be pre-diabetic and already be having these associations with fluid buildup in the body because of these vessels not being able to deal with that fluid flow properly. Okay, so starting to get the big picture of how your lymphatic system is going to work. AJ.
So basically, you get hurt, blood leaves your like veins and stuff like that, and then this will push it back towards the heart wall, so like filtering and making sure it's all good.
So before that fluid rejoins your circulatory system, we're going to filter it multiple times through your lymphatic system, including your lymph nodes, which we're going to talk about in a moment. But that starting to make a little more sense what your lymph system is going to do for you. All right, it's a separate system, and it deals with filtering everything that left your blood vessels. It has to all be filtered before it can rejoin your circulatory system. So if it leaves your blood vessel, it has to come back through the lymphatic system. And it's all driven by skeletal muscle movement. It is not driven by your heart pumping or any sort of, soft or smooth muscle movement, skeletal muscle movement thing. All righty, so we kind of talked about lymph. Lymph itself, that fluid that we're leaking out into our passages is lymph. And so lymph is plasma-like. It's got nutrients in it. It's got signal molecules in it. It's got white blood cells in it. And so that fluid is what's going to be filling your extracellular spaces, outside the cell spaces, and then causing the swelling as we're, responding to an infection or injury. And it all has nutrients for those cells to work, as well as, like I said, the signals between the cells, letting them know what they need to be doing, when, where, and how. It also controls how much fluid will be leaving your blood vessels. There's signals that control that as well. All of these proteins, all these molecules that your cells are making that basically cause any of this to happen. So when you hit your toe, it's not like a physical response leaking fluid in. What happens is when you have your toe and you injure cells there, cells in the area are releasing chemicals saying, oh my God, put fluid in here. We need help to start with repair. We need help to start with bringing white blood cells in and whatever. So your body's sending all of those signals because you have injured cells. The signals are what are allowing the lymph to move into that. So lymph itself is only going to move in one direction. Once it gets into the compartment, the only direction that that lymph is going to go is back to the heart. And it's all powered by your skeletal muscles. Here is a depiction of your circulatory system, roughly, right? And then your lymphatic system. And man, when I tell you, these are tinier vessels and they're pretty hard to spot. So when you guys take anatomy, there will be a few of these lymphatic vessels that you can identify. But for the most part, these are so small and hard to identify and hard to isolate within the tissues. You don't just see these things a lot. And I used to work in the transplant lab at Integris Baptist, and they would send us lymph nodes from our organ donors. And so what would happen is if somebody was declared as brain dead and they were an organ donor and they had otherwise healthy aspects of their body, then they would say, okay, this person's kidneys look like they're good to go to somebody that needs them and can use them. So they will send our lab that quote unquote dead person's lymph nodes. We'll get like 3 or 4 lymph nodes in a little bottle. How big do you think lymph nodes are? They're about the size of half of a pea. Half of a pea. Take a pea, cut it in half. That's the average size of a healthy lymph node. So if you're infected, you know how big they swell up. They swell up pretty big when you're infected. It's all those cells multiplying in your lymph nodes, causing them to swell like that. So normally they're really, really tiny. If you're a smoker, most of your lymph nodes will be black.
Ew.
Cute. Just like put it in perspective of what the lymph nodes are doing with you. Okay, so and so we would know if we got like, you know, kidney transplant coming in and somebody's lymph nodes were black or dark colored, we'd be like, oh, this person was a smoker. I bet that their lungs aren't getting donated, and sure enough, their lungs wouldn't be getting donated, but their kidneys might be fine. So we can still use their kidneys. And their white blood cells are still in the lymph node. We can still use their lymph node. It's just we know things about that person just by looking at their lymph node. So anyway, so your lymph nodes are actually really, really tiny, and you have thousands of them. And we have also tissues in your body that aren't really lymph nodes, but that have similar functions to your lymph nodes. And a lot of this goes in what we call associated lymphoid tissues. We're going to get into those too. So our organs that are associated with your lymphatic system, obviously, in your lymphatic system, you have the lymph vessels, and you have the lymph itself, which is the fluid, which is technically a tissue, right? But the main primary lymphatic organs are going to be your red bone marrow and your thymus. And so your thymus is where T cell maturation occurs, and we're going to talk about that in the next chapter. But your thymus is part of your lymphatic system, part of your immune system, and it's considered a primary lymphatic tissue because its sole purpose is for providing cells to your immune system. Okay? The red blood, your red bone marrow. In your bone marrow, what's in the center of all of your bones is the marrow, and it's soft sort of tissue. And basically what it is stem cells. And these are stem cells because they can become any blood cell. They can become red blood cells or any of your white blood cells. And they're called what we call hematopoietic stem cells. These hematopoietic stem cells will receive signals from the body telling them which cell to turn into and develop into. So this is where your red blood cells come from, where your platelets come from, where all of your neutrophils, your macrophages, your B cells, your T cells, they were all just hematopoietic stem cells in your bone marrow. And so that's why when we have somebody who has like leukemia and all their, like some of their white blood cells are just cancerous and they're going out of control and they can't be controlled, we can irradiate their whole body, kill all of the white blood cells in their entire body, and then get bone marrow from you and take your bone marrow, your stem cells, and put it into them. And now they've got new stem cells. And those stem cells can be trained and turned into whatever cells that their body needs them to be turned into, and we can replenish all of their stem cells that way. Really neat stuff, right? So all of your blood cells, all of them, red, white, or anything, all come from that hematopoietic stem cell precursor in your bone marrow. All right, next, our secondary lymphatic organs. Obviously, it's going to be our lymph nodes. These are areas where our white blood cells are going to hang out and where a lot of our filtering interaction is going to occur, our checkpoints. Then we have our spleen. Your spleen is really important for getting rid of old red blood cells as well as damaged red blood cells, but its other function is also going to be acting as a lymphatic tissue. So it works very similarly to your lymph nodes. But we've already talked about how your lymph nodes are filtering the lymph on the way back to your heart. And I already told you that the spleen filters out red blood cells. So clearly the spleen is seeing blood, not lymph. So it can filter out red blood cells. It's filtering out your blood. So the spleen acts as an accessory organ to filter out your blood separate from the lymph. And so it can catch microbes and white blood cells and things like that that are actually in your blood that aren't in your lymph. So it's acting to filter out your blood. Then we have your salt, malt, and galt. ALT stands for associated lymphoid tissue. So these is essentially functions similar to your lymph nodes with the filtering and interacting, but they're not in lymph node form. They're just associated lymphoid tissue. Diffuse tissue that has similar function to the lymph nodes, okay? So this is your skin associated lymphoid tissue, your mucosa associated lymphoid tissue, and your gut associated lymphoid tissue. So we have a lot of this tissue in these areas that have a similar function to your lymph nodes that aren't in lymph node form, which really all of these are. Okay. We went through these things, so I'm going to kind of click through them. If you want more information on these guys, you're welcome to. Your thymus is kind of in your upper part of your chest, kind of right here. And when you're a baby, it's pretty big. And as you get into adulthood, it shrinks to almost nothing. It's almost non-existent in most older adults. Most of your development of your T cells happens when you are young. You lose your ability to have the training and activation, the priming of your naive T cells, basically once you get to adulthood. Most of that happens in adolescence. So your thymus, which is involved in that, is essentially going to be shriveling away as you get older. All right, so those lymph nodes are bean-shaped organs that are very small, about the size of half pea, typically. And they're stationed all along your lymphatic vessels in order to filter out everything going through it. You have major aggregations of the lymph nodes themselves in like your armpit, in your neck, and in your groin. This is kind of obvious stuff. If you've been sick and you've had those swollen lymph nodes in your neck, that's swollen lymph nodes, that really is what it is. And so whenever we have that, we can actually palpate them as they're swollen. So you're actually reacting to an infection in your body. You're not really going to get that big if you actually have an infection. Typically, if we're just transiently interacting with microbes, they don't swell that big. They don't need to, because a lot of times your innate system will be taking care of it. And most of the time when your lymph nodes are swelling like that, it's because your acquired immune system is getting involved. We'll talk about how that works in the next chapter. All right, groin and your neck and your armpits. I don't know what else to say about it, but they get larger whenever we start having any infection. This could be a systemic infection, in which case a whole ton of your lymph nodes will be swollen. But if you're only having lymph nodes swollen, like in your neck region, you probably have an upper respiratory type of infection. It's localized to your head or your throat, maybe in your respiratory area, but it's a local or focal infection typically associated with that. If you're systemic, your lymph nodes will be swollen everywhere. So that can indicate what kind of infection you have. Again, the spleen is filtering out your blood, not your lymph, but it still has a similar function to your lymph nodes, just for your blood. We have our associated lymphoid tissues, our salt, gut, and galt, and these are basically just collection of tissue that have high concentrations of our white blood cells, particularly our lymphocytes. Talk about the difference between lymphocytes and other white blood cells in a moment. You take whole blood, which is like if I take a needle and put it in your arm and collect a tube of blood. So you guys know how there's different colors of tubes that they'll use for collecting blood. And you may or may not know what's in those tubes. Actually, probably most of you don't, and that's okay. But if it had something extra in them, our red top tubes that are just red top, it's just a plain old tube. It's just a tube. It doesn't have anything else in it. So our red top tubes, that's where you'll collect the blood, and we'll let it sit out on the counter to coagulate. So all of your platelets and all the clotting factors in there will be acting, and it'll clot your whole blood. So eventually, if I let it sit there for 30 minutes, you would take that tube that's been sitting in that rack, and you could turn it upside down, and the whole blood will just be sitting there. The whole thing. The red tube of blood that was just liquid blood, within 30 minutes should be solid. And so we can take that solid red top tube with the blood in it and spin it in a centrifuge. So all the solids will separate down at the bottom, and the liquid will be up at the top. That liquid that's up at the top, we call that serum. So the liquid portion of your blood, separate from all those clotting factors and everything, is called serum. Plasma is when we separate out your red blood cells from everything else. So what we do with plasma is, in order to collect plasma, we have to have anticoagulants in it. So we prevent that coagulation from happening. So we can prevent the platelets from getting stuck in there. We can prevent the clotting factors from doing their job. We can look at all the proteins and everything in your blood when we look at plasma. So if it is a color of tube other than red or tiger top, which tiger top, if you know what I'm talking about, it's the one that's red mottled with black on the rubber top. So that has a wax in it to just help separate the solids from the liquids. It's still serum. But plasma is when we add anticoagulants into it. So your green top tubes, your blue top tubes, your pink top tubes, all those guys have something in it to prevent coagulation. And no matter how long you let them sit on the countertop, they'll always be liquid, no matter what. You spin those in a centrifuge. Some of them have a little wax in the bottom, and some of them don't. But it essentially just separates your red blood cells from everything else. And that everything else is plasma. That's the difference between plasma and serum. Serum, we're separating out all your coagulated bits, and all the liquid, including maybe antibodies and things like that, will be in your serum. Plasma, we're taking just the red blood cells and separating it from everything else. Just the red blood cells from everything else. And the special anticoagulants that are added to those tubes let you separate it how you need it separated for whatever test you're doing. Yes?
So plasma is made of the red blood cells?
So plasma is the liquid portion of your blood. So plasma is everything but your red blood cells. Plasma is, you have red blood cells and then you have plasma. All right, that's essentially how it works. And so we can spin your plasma tube your anticoagulant treated tubes in the centrifuge, and you'll have red blood cells. This area that they call the buffing coat. It's a layer that settles on top of the red blood cells. That's where your white blood cells and your platelets are going to be. And then the liquid or everything else is going to be in the plasma portion. So it's the liquid. Treating something with the anticoagulant allows you to create this separation. If you let it clot on its own, all of the cells are going to be caught in what's the solid part. All of them will be coagulated all together. If you want separation of everything out, plasma, adding the anticoagulant allows you to do that. We talk a lot in class, we talk about how, oh, this is in the plasma. We donate plasma. I hate to break it to you, but treating your blood with anticoagulant. That's how they're getting everything that's good in your plasma out, right? And they'll deliver back to you your white blood cells and your red blood cells, and the things that would be in this and in your buffing coat. Because all they need is the liquid portion of your blood. It's the proteins that are in your blood and things like that, the nutrients, all that stuff's in your plasma. So if you let it coagulate first, the liquid is called serum. If you're adding anticoagulant, the liquid is called plasma. So there is a difference there, and it does matter, especially if you're doing certain tests. So that's as much detail as we're going to get into it with here. If you have more questions about it, I haven't go into it. Having worked as a phlebotomist for like forever, I can tell you how this stuff works. Also, another tip I want to get to you guys, if you're going into healthcare, whether it's human or animal, if you think that being a bad stick is okay because it's more important to be a nurse and be good at your other parts of your job than being a good stick, get over yourself. It is important to be a good stick. Learn how to be a good stick. If you think, oh, I'll just get through this and then I'll be good at the rest of my job. No, so don't be that person. And so you're going to draw better tubes and your patient is going to like you a lot more. You'll have better interactions with your patient and you will get more reliable data for treating your patient if you learn how to be a good stick. If you need help with learning how to be a good stick, talk to a phlebotomist, especially somebody who works at OBI, because they're using 14 gauge needles. The smaller the number, the bigger it is. Butterflies are 22 gauge. It's super tiny. If phlebotomists can put a 14 gauge needle in somebody's arm and their blood flows continuously from that arm for 10 minutes straight without any problems, then you should have no problem with the butterfly needle. There's no excuse for you. I don't care how sick your patient is. And I can say this because I worked at OBI drawing blood and I worked at hospitals drawing blood. I know the difference. And I'm telling you that being a good stick with one of those big needles made me a better stick at working with the little needles. I've stuck in hands, I've stuck in arms, I've stuck here, I've stuck here, I've stuck here. We weren't allowed to stick beyond the arms unless it was little babies and we had special circumstances. Let me tell you what's sticking with babies. We're going to be sticking babies. Good luck with that. You can't just tell them to be still. So what I'm telling you, like, can I just make a demonstration really quick? You will have somebody, yes. So if you were sticking a baby in its arm, you would have somebody holding their arm like with pressure, like holding the baby's arm like this. The mom would be holding the baby, and then a third person would come in with the needle trying to stick it while you're holding that all still. And that baby will be screaming and kicking and writhing about the whole time. Good luck with it, y'all. Good luck with that. Because I've done it multiple times. I'm telling you, just drawing a tube in a situation like that is hard. Getting an IV that operates properly, good luck with that.
How do you think you could IV in a baby?
I don't know. They have to give them sedatives, right? Don't they have to give them sedatives in order to like?
So my son had to have on his lip and his nose.
Sorry if I
So
hurt your arm. I didn't mean to hurt your arm.
basically what they did was they put it in his arm right here. And he kept trying to pull it out, so they had to put a bandage over it and a little
Yeah.
cut thing and put a bandage over that
That.
too, so that he could
So you
not.
couldn't pull at it. Yeah. So,
Yeah.
anyways, I'm just saying, phlebotomy skills, work on them. It's worth it. I promise. Don't just push them at the side. In the beginning, when you're just learning your skills, focus on your skills. But once you get to that point where you have your base skills down, work on your phlebotomy skills, guys. It is worth it. I promise. And having to rely on, if you're that person who's a bad stick and you're constantly having to call, you know, your charge nurse, or if you're constantly having to call phlebotomists, if you're working at Mercy, they'll have phlebotomists that can work the floors. It looks bad on you. That's all I'm saying. And if you're the patient and you're like, I have to call somebody else to do this stick, the patient is going to be judging you, right? Wouldn't you judge your nurse if they were doing that? I'd be like, does this person even know are they good at their job? I want the nurse that's good at their job. If they have to call the charge nurse to do their job, I want the charge nurse to be my nurse, not this person. Tell me you don't think that when you're a patient. Your patient will be thinking that about you. So be good at your job all around, not just parts of it. All right, so let's talk about your white blood cell. When you separate your blood, you have all of your liquid portion and all the proteins and the signaling factors in the liquid. Then you have the cell portion. These are your cells that are in your bloodstream. You have your red blood cells, you have your platelets, and you have your white blood cells, right? Your white blood cells are separated into two categories, the granulocytes and the agranulocytes. A granulocyte is literally defined by, if I look at it in a CBC and I take a sample of your blood from your CBC, your pink top tube, and I look at your blood in a microscope. And we do special stains on your blood. If we do the stain and I look at your blood and your cells have granules in them, little tiny dark dots in them, these are called granules. If they have it there, it's called a granulocyte. If it doesn't have those granules, it's called an agranulocyte. And that's literally how these are separated out. And as it turns out, these often relate to other functions that they have. The granules are often associated with materials that those cells can release out as signaling molecules, as well as for destroying targets. All right, so here's our breakdown. Our granulocytes fall into these categories. Two major categories of granulocytes that you need to know from my class. Your two categories of granulocytes, the ones that have the granules, the dark spots in them. The fills, it ends in the word fill, it's a granulocyte. And then one other, which is the mast cells. Those are your granulocytes. The fills and the mast cells. So the fills include neutrophils, eosinophils, and basophils. Our understanding of eosinophils and basophils and their function is incredibly weak. But we know neutrophils really well. Neutrophils are the most numerous white blood cells in your blood. 55% approximately of your white blood cells should be neutrophils. So 45% is everything else that's white blood cells. And so those come with their own numbers associated with them. But neutrophils are the most numerous white blood cells in your blood. And we know what they do. They are going to be involved in phagocytosis, breaking down things that are foreign and digesting them, secreting toxic substances to kill things outside of those cells. And that's really all they do. They do this sort of nondescriptly. And so they'll come and they'll see a PAMP and they'll say, oh, you're a foreign thing. I'm going to eat you up. And they just digest it out and crap it out everywhere. That's all they do. This is very non-discriminatory. And most of the time when you have an infection, you see pus, the white pussy goo that comes in with infection, that's mostly neutrophils. There'll be some dead bacteria and stuff too, or dead, skin cells and things like that, but it's mostly neutrophils. Your neutrophils are really coming to these injurious areas, coming to digest dead cells, coming to digest bacteria, coming to get rid of any debris or anything foreign. And that's what neutrophils do. They're very nondescript, very nonspecific. So that's neutrophils. Our basophils are very involved in inflammatory response. So they'll have some sort of phagocytic action, but they're not anywhere near as important in that as the neutrophils are. And then the eosinophils are involved in your parasitic response. So protozoa and helminths are your parasites. Protozoa and helminths. So eosinophils are made by your body to target that sort of infection. And I don't know if you've noticed, but Americans don't have a whole lot of parasitic infections. Don't have a whole lot of exposure to parasites. Not the protozoa and definitely not the helminths. So a lot of times our eosinophils are going unchecked. They're not being used what they're supposed to be used for. And so they're often get caught up in our allergic responses, which we'll talk about in chapter 17, what an allergy is and why it happens and things like that. But we think that this is due very closely to our response to basically not seeing helminths. Our body isn't being triggered. We're not seeing parasites. And so our normal response to parasites, it still exists in our body, but we have nothing that is training it properly. We don't see parasites. And so it overreacts to other things that aren't parasites. Talk about how that works in chapter 17. So that's your fills. Mast cells are basically something that reacts to things you've seen previously and start the inflammatory process when you see it again. Mast
memory cells?
Huh?
Memory cells?
They're not memory cells. Okay. So I'm going to talk about how they work. Again, this is going to be way more important in chapter 17. But essentially, when your body makes an antibody to something, certain kinds of antibodies will be held on to by the mast cells and they sort of decorate themselves with the antibodies. They don't make the antibodies. They pick them up from your bloodstream. So they are like, oh, the body made some of these kinds of antibodies. We'll put that on our surface. When those antibodies see the foreign thing, let's say it's pet dander. Your body might see pet dander as foreign and react to it and make an antibody to it. So makes that certain kind of antibody, your mast cell decorates itself with it. So the next time that you see pet dander, it's held on to those antibodies, anti-pet dander guys. And the mast cell recognizes when the antibody sees the pet dander and it releases all of its inflammatory stuff, including histamines and things like that. So mast cells are incredibly involved in your inflammatory response, especially in allergy. But they do other things in your immune system too. They have normal function in your immune system. It's just we see them really overacting to things that aren't immune related sometimes in our allergies. So that's our granulocytes. Our agranulocytes is basically categorized in two ways. The things that don't have granules. This will be your lymphocytes, and then our monocytes and their progenitors. A monocyte is what it sounds like. So it's like one cell, monocyte. Well, it has one big old fat nucleus in it and no granules. And so these guys are basically going to be floating around in your bloodstream, and they are general phagocytes. Once they get called out of your bloodstream, you stub your toe, it's going to send signals to pull monocytes out of the bloodstream. Once they get activated out of the bloodstream and move into a tissue, they differentiate into macrophages. I know you guys have heard of a macrophage. You might not know what it does, but you've probably heard of it. The macrophages are the heavy lifters as far as our phagocytes and then starting up your acquired immune response goes. So your neutrophils do a lot of dirty work. They're like the Marines when they get to a war. The neutrophils will come in and they'll just take out any of the bad guys. All right, and they're mostly just more or less nondescriptly taking out bad guys. The macrophages will get called in at the same time. So they're B monocytes, and they move out of the bloodstream and into the tissue, they become macrophages. If they're associated with the skin, they're called dendritic cells. So they're the same thing, they're just different versions of monocytes, right? So our monocytes and their derivatives, macrophages and dendritic cells, these are phagocytes in the tissues that'll get called in there in the inflammatory response. And they'll start eating up the waste of the neutrophils. So maybe the Marines were going around just shooting whatever they saw as foreign. The macrophages will come and clean up that mess and be like, okay, what do we need to be worried about here? Is this just all we need to be doing? Or do I need to start calling in more reinforcement? That's what the macrophages are going to do. So they'll eat up some of the bad guys that the neutrophils didn't get to. They'll also be taking the waste from the neutrophils and basically analyzing, is this a sign of a bigger threat? That's what the macrophages are going to do. And so they're going to send out signals from the body, basically saying, okay, let's go to the lymph nodes and warn the people at the checkpoint that we need to call in the bombers or the tanks to bring in reinforcement into this area. And lo and behold, in my example, the bombers and the tanks, they're the specialists. So you call in a bomber and it's going to come and attack this specific guy or the specific group of bad guys. It doesn't just come in and bomb the whole area, everything in the area. It knows who they need to bomb. And so when you call in a bomber, you're only calling them into this specific place or this specific time for this specific bad guy. Those are your B cells. So your B cells are the bombers. Your B cells, the cells drop the bombs. And what are the bombs? Your antibodies. Okay, so your antibodies, your B cells will get, your macrophage comes and eats the crap that your neutrophils pooped out, and it brings it to the checkpoint. And the B cells at the checkpoint will say, okay, well, shit, like I see this and I need to react to that. And so here's antibodies against that specific molecule. And the bombs will go and find just those bad things. That's essentially how your immune system is going to get triggered. So the macrophages are the go-between. They're the whole kind of governor of how your immune system is going to respond to something. They might just clean up the mess and go show it in the lymph nodes, and everybody be like, no, that's fine. You don't need to worry about this. But sometimes it's going to see that and be like, oh, okay, well, crap, we need to drop the bombs and we need to call the tanks into those areas. In terms of your T cells. So your lymphocytes are your B cells and your T cells. We're going to talk about your lymphocytes and their functions and what antibody actually does for us and all that stuff when we get into the next chapter. Okay. So those are our white blood cells. All right. Your mononuclear phagocyte system is basically the network between your cells and tissues that allows your white blood cells to move around. Really all it is. It's like a highway between your cells and your tissues that allow your white blood cells to migrate into those compartments. So in order to get that liquid out of the capillaries, that doesn't move into the next cells, it moves into the space between the cells. And your white blood cells that come with that liquid into that injured area, the white blood cells that move out of your capillaries into those areas, they're going to move around by the mononuclear phagocyte system. It's a passageway to move around in the body. Again, your monocytes, big, one nucleus, they don't have any granules in them. These will be able to move into tissues and become specialized to those tissues. So once they move out of the bloodstream, they'll kind of basically function in those tissues for the most part. So we have Kupffer cells in your liver. These are basically essentially differentiated monocytes specific to your liver. You have alveolar macrophages in the alveoli in your lungs. Talk about how tuberculosis lives inside of those macrophages, survives that. We have Langerhans cells in your skin. So these are dendritic cells that have differentiated in the skin. And we have in the brain special monocytes that we call microglia. So they have a reduced immune impact. So they're not causing as much damage with inflammation as what regular white blood cells would do. Because your brain can't regenerate, right? Your nervous system can't regenerate. Cytokines, this is the term for proteins and signaling molecules that basically guide the whole process. So these are special chemicals released by cells. So you injure your toe, and because now suddenly you have broken cells that have been broken open, it causes signals to be sent by nearby. So you break your cells, cut your finger with a paper cut. There are cells that got damaged there, and they're going to release chemicals saying, I got damaged. And then cells nearby will be like, oh my God, somebody got damaged near me. We need to call in help. And so those are our cytokines. Those call for help vibes. They're just molecules that are telling the immune system what to do. Those are cytokines. The word for that. So these will be things that are pro-inflammatories that encourage inflammation. We also have signals that will be anti-inflammatory. So once everything's starting to die down, we'll have signals sent out to lessen our inflammatory response. We have vasodilators and constrictors to control blood flow into that area. When you first get injured, let's say you cut your finger when you're like chopping up your food for dinner, you cut your finger, your body will vasoconstrict to prevent microbes from getting outside of that area that you injured. So it might vasoconstrict your flow out of the area, but vasodilate the blood into the area. So that way we can have blood flow into that area, and you'll see the swelling from all the liquid and the blood in that area, but we'll constrict the flow out of that area to keep everything confined in that compartment so that microbes that were introduced to the injury site don't get out of the injury site, but we can still get white blood cells there. So signaling molecules will control that, vasodilators and vasoconstrictors. And we also have growth factor cytokines. These are signals that are sent to help trigger calling in and growth and multiplication of your lymphocytes. Okay, as it turns out, you might have a B cell that recognizes the spike protein of COVID. You have one in your entire body, one B cell that will recognize that spike protein. Once that B cell, which was waiting happily at some lymph node somewhere, waiting for the signal from somebody to come in there and basically tell them what's going on, that B cell, I don't know how to tell why so fast am I talking. So that B cell will effectively get triggered and told to make certain antibodies that will cause it to multiply. So you have one B cell, and once it gets activated by that antigen, would multiply out and create multiple B cells. So you only have one until it gets activated, and then it'll multiply out. That's why your lymph nodes swell, because you're multiplying that one B cell a whole bunch. And you might recognize several things all at once in an infection as well. So that's the idea with that stuff. I want to touch on something important. I will come over and review some of this stuff in another lecture because I have run out of time. All right, lysozyme is an enzyme that breaks down peptidoglycan that's made by our body. Phagocytosis, when our white blood cells eat things up, our main phagocytes are going to be your neutrophils and your monocytes and macrophages. Those are your main phagocytes. Other things might have some phagocytic function, but these guys are very key in phagocytosis process. Neutrophils, and then the monocytes and their derivatives. So you can kind of look at it as two, because remember, macrophages and dendritic cells are derivatives of the monocytes. They're just specialized. We'll go over the steps of phagocytosis later, but it basically involves chemotaxis. There'll be signals sent by nearby cells that'll call the macrophages or phagocytes into the area. That's chemotaxis, responding to chemicals in the area and then moving accordingly. We'll ingest the thing, and then we have ingestion into something called a phagosome, And you have your lysosome, which remember we talked about has toxic compounds in it. When the phagosome, which is the vesicle you ate it up into, and the lysosome, which is the toxic vesicle, when those things merge together, it's a phagolysosome. And then the digestion occurs over a process of about 30 minutes. That destroys the thing, that's the destruction. And then we'll just excrete the waste product out. All right, we talked about PAMPs already. So this is our signs of inflammation, our redness, our warmth, our swelling, and our pain. So they also have Latin words that you guys do need to know. Rubor, calor, tumor, and dolor. Tumor, like you think of a tumor as being a growth, so it's swelling, it's fair enough. You don't know Spanish, and these words might not be intuitive. Rubor is redness, like rubies. Calor, like calorie. Calorie is literally a measure of the amount of heat given off by burning something. That's a calorie. So calor relates to that, it's heat. Right? And then dolor is pain. We also have loss of function in some cases. Whenever you have enough inflammation going on, that can be a sign of inflammation. And then, of course, chronic inflammation. If you have a whole lot of this going on, and we're constantly signaling for your immune function, this can lead to damage over time. And if you have prolonged effect of this, it can lead to pretty serious damage and effects of that. What else? We kind of talk about a lot of this stuff. So that fluid flowing into that extracellular space will lead to swelling. That fluid is called exudate, and edema is what you see when we have that compartmentalized. So swelling of a tissue from the fluid moving into that area. White blood cells, when they're moving out of your capillaries, we know it makes sense to think about fluid flowing out of a capillary and into a compartment, leading to edema, and all the signals that come with it. But the cells also come out of your capillary. And then they move out of your bloodstream and into the tissue is through a method called diapedesis. So diapedesis is that process of them squeezing through. So these cells are made to be basically squeezed through your capillary walls, like the cell junction. They can just squeeze through there. All right. And we'll talk about fever and stuff in the next one. I don't want to get too much into anything else because we have to go to the lab. We'll pick up from fever next time. I hope we're starting to get the big idea of how your immune system works, and a lot of this will come into play later on.
Good thing I left the one blank page for my next chapter.